To build a primary immune system, mammals rely on sheer genetic volume. Developing B cells in the bone marrow generate their defensive diversity through a process of combinatorial assembly, randomly shuffling and recombining massive families of V, D, and J gene segments. Birds operate under a different genetic reality. For their immunoglobulin light chain, the avian genome contains only one functional variable, or V segment, and exactly one functional joining, or J segment. This diagram illustrates the mathematical limitation. If a bird relies on standard combinatorial rearrangement with a single VL1 and JL segment, pairings hit a biological bottleneck, maxing out at roughly 1,500 total combinations. A vertebrate requires an immune repertoire of millions of distinct specificities to survive exposure to natural pathogens. Yet, as the math here indicates, the rearrangement of the avian light chain yields a total diversity of about 10. To survive, the avian immune system abandoned combinatorics. Instead, birds employ an enzyme known as activation-induced cytidine deaminase, or AID. Operating in a primary, strictly antigen-independent setting, this enzyme engineers diversity from the top down. Large recombining gene families are not a strict biological requirement for generating a primary immune repertoire. Highly localized genetic engineering achieves the same goal. Looking at the lower section of this genetic map, we find the architecture that makes avian diversity possible. Located directly upstream of the functional locus is a cluster of 25 pseudogenes. They lack promoters and cannot be transcribed, rendering them a static, read-only database. During transcription, the DNA unwinds, exposing single-stranded sections. The AID enzyme actively targets these strands. Upon locating specific WRC motifs, AID deaminates a cytosine residue into a uracil. This leaves a localized mismatch in the DNA duplex, immediately triggering the cell's standard error repair pathways. Paralog proteins, molecules like XRCC2 and XRCC3, arrive to resolve the mismatch. They refer back to the read-only pseudogene database, copying entire homologous sequence blocks. These imported blocks patch the error, permanently overriding sections of the rearranged junction. Up to 300 nucleotides are swapped out, completely altering the receptor's antigen binding specificity. By repeatedly copying and pasting code from a silent database, somatic gene conversion allows a single functional gene to generate a broad, highly customized repertoire. This rapid, highly targeted genetic diversification cannot happen in a standard hematopoietic environment like the bone marrow. It demands a specialized, dedicated anatomical space. This schematic tracks the physical journey required. During embryonic development, pre-bursal B cells exit the blood circulation and track a specific CXCL12 chemokine gradient, crossing into the mesenchyme to colonize the avian Bursa of Fabricius. To cross the basement membrane and successfully colonize the inner bursal follicles, these developing cells must pass a strict developmental checkpoint. They must possess a functionally signaling B cell receptor complex. Colonization is strictly mechanically driven. Researchers engineered a chimeric receptor, fusing internal B cell signaling domains to an external CD8 structure. Both the native receptor and the chimera undergo continuous internal phosphorylation. This basal signaling occurs even without an external target. Those chimeric cells successfully migrated into the bursal follicles, initiated rapid proliferation, and engaged the AID enzyme for gene conversion, performing identically to native cells. Because colonization and diversification proceed normally with a receptor completely incapable of binding outside molecules, the earliest stages of avian primary repertoire generation must be blind to external antigens. It relies entirely on internal, mechanical basal signaling. Immediately after hatching, the bursa's internal anatomy reorganizes. The previously uniform space of rapid cell proliferation segregates into distinct inner medullary and outer cortical zones. On the far right, notice the follicle-associated epithelium, or FAE. After hatching, specialized tufts connect the internal lymphoid compartment to the gut lumen. They actively transport environmental molecules from the gut into the medullary compartment, where they are presented by follicular dendritic-like cells. With this anatomical shift, the rules for cellular survival change. The basal signaling that carried these B cells through embryonic development is no longer sufficient. They must now achieve specific receptor ligation with these new environmental antigens. We see this clearly in those experimentally engineered chimeric cells. Because their altered receptors cannot bind external molecules, they are blind to the environmental antigens now flooding the bursa. Post-hatch, those cells rapidly undergo apoptosis. Native B cells whose newly mutated receptors successfully recognize a gut-derived antigen are selected to survive. They migrate across the junction into the follicular cortex, where they proliferate and finalize their genetic diversification. The bursa shifts from an isolated genetic sandbox into a highly calibrated environmental sensor, integrating local gut flora to select a primary B-cell repertoire uniquely adapted to the bird's immediate surroundings. Once diversification and selection are complete, the bursa continuously exports roughly 5% of its newly generated B cells out into the peripheral bloodstream. Notice the migration pathway into the spleen splits. Emigrating from the follicular cortex, the vast majority form short-lived, naive B cells expressing the LT2 antigen. The remaining 10% lack LT2 and live significantly longer. These represent the specific cells successfully selected by gut antigens. In mammals, producing a primary immune repertoire dictates a reliance on massive recombining gene families residing in the bone marrow. Birds provide a stark contrast to that mammalian biology. Starting with a single genetic sequence, they utilize targeted enzymatic mutations to build a diverse database of receptors, strictly curated by environmental exposure. The avian immune system demonstrates that evolutionary pressure can solve the exact same problem of primary immunological diversity through a completely inverted set of rules.